Hold on. Hey, see that?
You're wearing a sweater.
Right there. Mary Ellen Moffat. She broke my heart.
Right. Well, good afternoon. It's good to see the stragglers here, the diehards. It's an amazing conference, Stuart. If you can get people coming for workshops on Monday and they're still here on Saturday afternoon, that's pretty darn good. No, you guys are super. And I was actually speaking to one of the itinerant speakers, who I hope will convert to a regular speaker earlier today, and he was saying that it's a really great community. People who care about this topic and are coming and talking about it and thinking and working hard. It's so again a tribute to Stuart and to you for coming and thinking about this, and we'll work our collective consciousness. So this afternoon, let's talk about where did it all come from. So our session is entitled The Origin and the Evolution of Life and Consciousness. And we're going to start with Dr. Bruce Damer of UC Santa Cruz on the origin of life and consciousness. Dr. Damer.
Thank you. I want to thank the two Stews, Stuart Kaufman and Stuart Hameroff. Stuart Kaufman had recommended me last year, and he was doing more of an origin of life theoretical biology approach, and that's how I got put together with you, Stu. And thank you very much for this kind offer. What you're going to see today is actually the first time this work from science has been presented outside of our community. So this is the first time I'm presenting this in an audience where it isn't two-thirds chemists. So this is quite a little bit historic. I was writing to our team last night saying, this is it. This is where we launched this thing. So thank you again. So what I'm going to do also, to provide you some grounding, I'm going to pass around an artifact. Spend a few seconds with it. What this is stromatolite, some of the oldest evidence for life on Earth. And I'll be showing you another during the talk. So feel free in the next 30 minutes to make a connection with it. So off we go. So this is a novel approach to the origin of life. This is the first end-to-end scientific theory, hypothesis, testable in the lab for the origin of life from basic molecules to cellular life. It doesn't go beyond that, of course, but it gets you to that point. And it possibly has implications for consciousness and everything else. The disclaimer, this wording was assistance from someone here at the meeting helped me with the wording so I could make it more fitting with what you guys are working on. So try to treat this abiogenesis stochastic cycling implications seriously, and you'll see the cycle because it's supported by empirical evidence. We found an engine that can boot stuff up in chemicals, but where it meets the hard problem, the Chalmers hard problem, please treat this as playful speculation. So what you're about to hold in your hand and what I'm holding in my hand here is new for science. This is some of the oldest evidence for life on Earth, 3.48 billion years old, that was discovered in the Pilbara last summer field season. And these little red nodules are stromatolites. They're layers laid down by organisms which sequester sand grains together. And then they stack those and they grow up on top so they can keep access to the sun. That's the whole name of the game. And they built these rock textures or fabrics, all cones and domes and you name it, and flat pieces. And this is the sole story of life on Earth for just about 90% of the time. And this is still the bulk of the story of life on Earth. We're this rarefied thing called complex animal life that popped out maybe for about 2% of Earth's history and will go away. But microbial community is the story of life on Earth pretty much and by bulk. So I'm going to take up not your hard question, but 3 other hard questions that might be related. how did the non-living world, how did non-living matter animate itself into the living world? That's one. Why did life bother to come into existence, or did it have a choice? There's your free will for you. And this one, which is very speculative, and I'll be quite happy to get critiqued by this group. What is the source of all, an arbiter of all experience? Which somewhere in there is consciousness. So a little bit about me. I have two career paths. One is working with NASA on spacecraft. So there's me at NASA. I helped NASA create a whole new architectures for missions to the outer solar system, but also to the asteroid belt, if you will. This is my design for a human mission to an asteroid. It's the first reference design. I've done about 25 projects for them. This is asteroid capture in a balloon structure and manipulation with gas called Shepherd, which can also make biospheres and do mining and get fuels. And this single invention by a team of 3 of us has the potential to open the solar system. So this is our current initiative, is to find a government program or government private to start running demonstrator flights and try to prove this technology to allow human beings to extend the biosphere into the solar system. So that's one aspect of my work. We're now working on site selection for the next rover. This is the Mars 2020 rover. It comes down on a sky crane. Its primary mission is search for life. It's the first NASA mission to search for life on another world, not just do the geology primarily or find the water. And we're arguing with NASA, we're debating, we're trying to convince them to go back to this location. This is a picture taken by the Spirit rover about 8 years ago of part of a rock outcrop, which was a hot spring that could preserve life just like this, about the same age. So we have a shot at finding a life on another world if we go back to Columbia Hills, which is where Spirit is sitting there right now. So rolling back the clock, how did I get interested in origin of life? I was raised in Canada, in Kamloops, British Columbia, Canada, and I love to walk in the nature. And I love sagebrush because to me it was like little forests. Like you're flying above forests when you walk through sagebrush. They're like miniature oak trees. And I was walking out in the spring of 1976, and I bent down, and there was this beautiful Mariposa lily coming up through the ground that had just been frozen. And my little 14-year-old brain just flipped and said, how did this structure, how did this beauty emerge? This is a beautiful plant, it's beautiful, and there's a bulb or a seed somewhere down under the soil that has instructions to do this. And then I stood up and I realized all the plants are running some kind of instructions. And then my brain sort of went into hyperwarp speed, and I went back and said, well, where did it all start? Did it start with one seed or one instruction? And then I thought, hey, I'm a nerdy kid. I'm a nerd kid in a town with no computers, but I'm a nerd kid, and this would be a great problem to work on my whole life. So that's when I committed to working on this problem. And I learned about this fellow. I was reading about Albert Einstein, that at 16, he had thought experiments. He actually had them even earlier, but he had one where he's running alongside a beam of light, and he sort of sees in his imagination, it's sort of a delivered vision this guy got at 16 about the compressing of the waves of light and things that led to special relativity. So I was on the verge of thinking, I could do that when this happened. And I drew this out. Like in my mind's eye, right in front of me, a thought experiment, a Gedanken experiment started happening. And it was a bundle of molecules. And I didn't even know what molecules were. I knew they were kind of things. I'd read some chemistry articles. And they're moving. And I'm formulating a question. You can ask thought experiments questions. I'll ask this one. How did simple molecules self-assemble into things that could do things on their own? And instead of getting that question out, it asked me the question, said, figure out how we made a copy of ourselves. Figure out how we made a copy of ourselves. And my little brain flipped and said, well, if you have a TR-7 automobile, it's made in a TR-7 automobile factory, which is a bigger machine, and I don't see a bigger machine here, so it's impossible. And the little bundle of molecules winked. And that's what I've been working on for 30 years. So here I am 40 years later on the trail of it. So this is me at Bumpus Hell, which is a hydrothermal field up in Mount Lassen National Park. It's actually a very dangerous place to be standing because Mr. Bumpus lost his leg there 150 years ago. What I'm doing is putting a slide tray into a fumarole vent, which has gases coming out. This is the slide tray, testing the theory that just vent gases alone with humidity could allow us to polymerize an important biopolymer. And it worked. This is the first time it's been done in the field. And this is our gel. And if you see that little blob there, that's RNA. We made RNA without enzymes in a fumarole vent environment. Because our lab goes to these environments, tries to do it in the messy chemistry of nature, for goodness sakes. This is what we use in the lab to do that. It's a device that cycles around 24 dishes between hydration and dehydration stations, and we have a 90 Celsius atmosphere. There's our hydration station, our dehydration station, and we put in reagents that are the building blocks of life, basically. And we can make polymers. We can make the building blocks of life without using life. And this is the first clue to how to do this. Here's our evidence, because our colleagues are very skeptical. They're chemists. They're incredibly hardcore materialist people. So you have to really do this thing right. So here's the gel electrophoresis, and those black bands on the right are our product. We invented a technology called nanopore sequencing, and we ran it through our own device, and it counts the base pairs as they're going through a pore and gives you little blockades, and you can actually do digital, like analog nature to digital directly. It's a really cool technology, and it worked there. And we blasted it with X-rays to see the stacking order. And then we discovered that we could get these polymers, these biopolymers, inside vesicles, inside lipid. The membranes that actually float in from space, actually through dust, they come in and they land on the early Earth, and you got your membrane supply, you've got your amino acid supply, and some of your building blocks are coming in from space. It's kind of a panspermia, but it's not a fully formed organism, it's just a building blocks. But we proved now, it's pretty much taken in the field, that we can grow polymers this way through basically drying things down and letting the water go away, and you can make these things. It's like nature's way of making biopolymers before life took over. And they can become encapsulated in these containers. And then you can subject them to selection by their trillions. Now let's take a look at how this works. So that's all the chemistry I'll show you. Let's go to a metaphor everyone in this room understands, computers. So if you're like me in the mid-'70s, your first computer might have been this clunking thing with switches on the front with a paper tape input. It wasn't quite that bad for me, but what you had to do is punch the program into paper tape. This is how Microsoft started, which was the basic loader to boot load, and then to load the basic language to then run real kind of language. Well, what if you started with a random puncher, but you had an infinite spool that made just zillions and zillions of program tapes, just trillions of them. And they went into a reader, and the reader went into your primitive computer. This is my Altair 8800. And it has an energy source, and it cycles. It just try to run those programs. And there's this little processor, and there's this little instruction. And they either crash the system, it goes into the crash trash, or it runs, play again. And you get program A. Program A can go back into the random feeder and get added to with, let's try B with that, and C with that, and D with that. Let's just try to extend that. Well, over time, you'll develop more functions. You might evolve a computer that will now have a screen and a keyboard if you're evolving both together. Keep going and you might get to a laptop. Keep going and you might get to your iPhone. So this is a model for evolving software and hardware together. The only thing different is that you're doing it randomly, so it takes millions of years. So we hire a cheap form of biology called engineers to do a slightly better job than the random puncher. And we get our programs written and things like that. There's no engineers in the audience, right? So this is an inefficient way of booting up a system. But this was perhaps the only way that nature had available to it, but it works. This method works. So let's replace this system. Let's go out into the wild and try to find in the world where we could do this. How could nature do this? Well, it's quite simple. The polymerizer is membranes and organics all smushing together, coming in, concentrating in pools, and polymerizing. That's what I was doing in Bumpus Hell. I was just doing that. So it makes polymers, which are your tapes, your punch paper tapes. Those are fed into a computer called a warm little cycling pool. There's our Charles Darwin, who in 1871 said, I think life started in some warm little pond somewhere. And he was extremely prescient. This one little paragraph is extremely right on. Absolutely. So as a source of energy, it's pumped by a geyser system. So what are your programs? They're called protocells. They're random collections of polymers in random little bags of things called lipids, lipid membranes. And how does your processor work? It's Charlie again, natural selection, a form of early natural selection where he says, oh, that one dislocates and blows up, and this one's fine. This one's more robust because the polymer did something to stabilize. And it kind of like microtubules or cellular structure might stabilize the membrane. Oh, that one can go forward and go again and again and again. Same system. So we put it all together, and this is artwork done for Scientific American, which will be in an article in August issue to announce this to the general public. Hot spring origin of life. So here's the big picture. You've got your landscape 4.3 billion years ago, called the Hadean period. You had your volcanoes, your Fred Flintstone volcanoes, and your dirty atmosphere, and your chocolate-covered oceans, and or colored oceans, and it's all infused with iron. That's why this stone that you're handing around has worked its way around is so heavy. It's full of oxidized iron. So we synthesize in the solar system all our building blocks, as I was telling you before. We accumulate them on land in little pools, get our organics, until they get concentrated enough to do chemistry. Because if they go in the ocean, they diffuse. So that's why the field's turned away from an ocean origin now. Just can't do it. You can't get chemistry to work. That's very, very difficult in a diffusing, a pregogene type environment. So then your organics are floating along. They're in their pools, and you're forming membranes and all kinds of goodies. And they happen to flow down into this optimal pool that's getting filled and draining and filled and draining, or filled and drying and filled and drying. And that creates a pump, a natural pump, to pump these polymers in through their film phase. This is where they're being written like paper tapes. That's where they're being tested by Charlie Darwin's idea. And then they accumulate in the bottom in this gel. That was our big insight. This is our big insight because this gel ends up becoming the common ancestor. It's a new idea for science. So once this gel it's just a smush of bubbles that have learned how to adapt and grow. And once they're tough enough, they can get washed downstream. They can go to other places. They get blown by wind if they're dry. And they start evolving and adapting. Then they start sharing early genetic functions, and you get the roots of the tree of life. So we found the roots of the tree of life, which is a cool thing. And they start adapting to tough environments like the seashore. Lacustrine environments, this is estuarine environments, and salt just blows you up. If you eat a whole bunch of salty potato chips, you got to go drink water, right? So we have to learn how to get rid of salt. That's why another reason you can't have an origin in the ocean. You need advanced technology to deal with salt. And once they get to the seashore, they've got all this sand washing over them, so they have to cement those sand grains together, and they create stromatolites at lake shores and at oceans. This is a model for the origin of this that was so dominant in Earth's history and will dominate Earth after we're gone. So here's a little view from below. So here's our little hydrothermal pool. Here's our cycling system. And really, it's all about the cycling. You have to turn the wheel and go through transitions to do this kind of a trick. It's all about the cycling. And you eventually get the progenote, the boot up phase of life, which is now becoming a field because we have a model to make these in the lab now. We can grow these in the lab, and we have sort of a challenge to different teams. You can grow these progenotes, and they can start booting up the functions of biology. Caveat. This is proto-natural selection for people who study this, and it's determined by physics. It's thermodynamic efficiency and statistical mechanics running, and what it does is pushing entropy out of the system and pulling syntropy into it. And it's doing, for those of you who study this kind of things, this is Stuart Kauffman ideas, it's doing stochastic hill climbing through noisy space till it gets to the adjacent possible. That's a Stuism, right? So, but biology comes into this system which is acting like biology. We got it for free, and it says, oh, I'll replace that with this. Oh, we found a function to make a pore. We found a function to do polymerization without drying down. We'll replace that. We found a way to make peptides and proteins and more. And so basically it's booting up an OS. This system is capable of booting an OS. And eventually this keeps running long enough, you get this. And what happens when you get a beautiful rainfall in California, it's been dry for a while, you get an explosion because you have a three-phase system. You have the dry, dry, dry. What's happening in dry? When systems are drying, the plants are getting ready to survive the dry season. So they put all their important polymers inside little capsules called seeds that aren't subject to hydrolysis, and then they lie around and they get blown by wind, they get washed by water. A rainfall event comes, it's a violent event, it distributes things, and then the moist period is when they start their metabolic process. They start sharing, they start generating. And then they get ready to make those polymers again in their punch paper tapes. It's the same thing. So we found a natural setting that is still patterned like a hand and glove onto the world that's around us. It's still patterned on our bodies. We grow and die based on this pattern. This is what made biology, at least purportedly. So 3 hard questions. How did non-living matter itself animate itself into the living world? Cycling. No need for a crater, no need for consciousness or any other action other than the physics of the universe. Why did life bother to come into existence, or did it have a choice? It just emerged given the right conditions. So in our quest now to try to find life on Mars, I did a presentation for the JPL and headquarters and big group in Pasadena. I had to do the first talk on the origin of life on another world, as in 12 minutes of terror, just like when the vehicle's coming down, they call it 12 minutes of terror. This is my 12 minutes of terror. 300 people in the room, mostly geologists again. And literally, hot springs, life emerges in this model. And unfortunately, for that life, if it did, Mars is dying. So Mars is warm and wet, and then it starts to die. And about 500 million years in, horrible things are happening to Mars. It doesn't have a magnetic dynamo in its core, so it doesn't have protection of a magnetic field, and there's an immense amount of atmosphere being blown away. And by 1 billion years in, the oceans are starting to dry up and sublimate and disappear. So if life is on Mars, it's like, ooh, this is not a very good place. I have to go for refuge, and that means through the pipes of the hydrothermal field, underground to the salty, wet rocks, and that they're stuck. If there's life there, it's stuck. They can't evolve very much forward. They're stuck in stasis until the sun, goes through a red giant and destroys the solar system, or the inner solar system. So it could just happen everywhere on these rocky worlds that have exposed land masses, enough feedstock, and enough cycling that came from the cosmos. It just could be happening. But it's extremely rare to have complex life. Totally, incredibly, impossibly rare to have complex life. But you've got trillions of exoplanets. The thing is that in order for us to get complex life, these guys had to, the reason they're red is because they're generating oxygen and they're fixing the oxygen in a process called rusting, and they're dropping the oxygen down to take it out of the atmosphere and the oceans and the water bodies. The iron is basically getting sequestered, because if they can't do that, then they can't put free oxygen into the atmosphere. So they have to fill the buffer first, and that takes 2 to 3 billion years to get an oxygenated atmosphere. And anything can happen to a planet on that time scale. Big impactor resets it, it dries down, et cetera, et cetera. We were fortunate. We went through snowballers. We went through, and we got to oxygenation. Then we got to complex cells. To get to us is extraordinarily rare even after that. So the hard questions, this is the one that's maybe pertinent to this group. What is the source and arbiter of all experience? And here's where I'm going to leap off from this platform. But it's an interesting platform, because if you unwind life to its boot up sequence, you can learn about what runs the living world. And what we've discovered is 3 properties that might have a bearing on what you're working on. And if we can discover those boot up properties, we may discover what consciousness is within the booted up OS. So I had a dream one night, and I dreamt, I said, wow, we're like, at the moment now, we're at the boundary between physics and biology. And for years I've been doing a dance with, I call her, Madre Nature, and saying, I'm getting close. 10 years ago I said, I'm getting close, and I had my box, which was my giant computational simulation engine I built. And she just laughed, a belly laugh. I will not divulge my secret to you with your puny box. So I switched to chemistry, because the computer is just woefully inadequate to do this kind of work, so we had to go to chemistry. Now we're getting really close to this. We're getting to the point where we may be synthesizing DNA through self-assembly. We're getting real close, we think. So Mother Nature, so I was back there, she's got her, her frock on, and she's saying, I'm making the cookies, but you're not going to get any, that kind of thing. So this is where I'm doing all my thought experiments, at this boundary. So one night I had a dream, and I saw the pure plane of physics, moving, moving, moving. And then suddenly in the plane opens this divot, kind of like if you're doing golf and you open a divot, a sort of a pocket. And I looked closely, I said, what is going on? Why is it pushing physics back? And I look in there, and there's something going on, and it's the usual protocell, but something going through the membrane, little molecule there, hitting something there, which decides to do an action. Boop, 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 one of Stu's autocatalytic sets. And I realized if this was not in the membrane, and this didn't pass through in a way, it would never happen. So the story of this part of the dream was biology takes improbable events and does this magic trick to it. It takes them and makes them more probable, more probable, more probable than it makes them actual. Kind of like quantum mechanics is supposed to do. Don't ask me, I know nothing about quantum mechanics. So it's got this property of probability tweaking, and it's a massive scale. Now physics has the same property, but it takes a long time. It's a cumbersome process. So just after the formation of the first stars, you're not going to see gold atoms anywhere in the universe. They're just impossible because the building blocks aren't there. But after, a few billion years, gold atoms can emerge because there's heavier building blocks been made by stars. So it does the probability thing too, but it's just really cumbersome. But when life starts, all kinds of things happen. So then I was shown in the vision 2 protocells next to each other, and there was a dip in between them. And the dip was deeper than I thought it would be. And the dream said, watch how this works. And there was stuff flowing back and forth between the dip. It said the connections are deeper than we could suppose in the protobiology, in the way to biology. The connections are deep. Then it said, are you sitting down? Because these dreams always say, are you sitting down? I always say, I'm lying down, because it's going to scale up. So it scaled up, and it started scaling up to more and more protocells. And like, whoa, this is a huge system. Just this alone. This is now doing more improbable things, boom, 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 and it's talking about them. Hey, I just did this improbable thing over here. Oh, I did one over here. I'll share it with you. Improbable thing, boom, boom, boom. This is a powerful system. This is the booting up of the progenote world. We're missing a piece, though. So as it scaled up, these Cartesian plots occurred hanging in space, and I saw these curves, probability, engine going up, connection going up, and then there was this third curve. It was like, what's the third curve? And then it did a funny thing. The dream did a funny thing. It shook the system, and I saw, our good old friend, informational polymer, appear, and this light poured out of the system, and it reflected back. Oh, that's pattern. It's invented patterns called information. It's like a patternome, and that's the third element. So then my little brain started rolling its dice, and I realized these are deeply connected. There's a probability engine operating, which can't be done in physics. There's a connectome operating, not a neural connectome, but just chemical messaging that can't run in physics, and a patternome, which is the first little oligomers of genetic sequence. And it's like these systems talk to each other rapidly and tightly, and they scale super fast, and they create something super huge. And so even in the Progenean, even at 4.3 billion years ago, when you have sludges everywhere, they're barely alive, and they can't even divide themselves, this is a massive, powerful computational, expressional probability tweaking machine. before even the formal transmission to life. So what I would put to you or offer to you is that these properties are all you need to explain all phenomena. These three interacting properties, and they generate all observable phenomena. They might create all experience if they're like apps, running in this OS called life. And human consciousness, primate consciousness, which is very specific, different from earthworm consciousness, samples only a small fragment of this. And we sample it by holding a mirror up to another piece of broken glass, and we're able to sort of see something. But it's all different for all different people. An autistic person would see a different way, and all people culturally see a different way. An Aboriginal person would see it a different way, feel it a different way. And it's all different, it's all relative. So the message of the dream, so usually what happens in these dream experiences, at the end I get my shirt collar grabbed, like, you listen and you listen good. I try to get this message through. And the message was, you primates, you monkeys are only at the very beginning of sensing and studying this field. You're just starting. That you should be wary of naming it and modeling it prematurely. It's huge. Because if you come to a molehill and say mountains, we can characterize them, and then there's a hills there, and then there's Himalayas there. You're going to never go to the mountains, right? You're going to stick with this old molehill model of everything. The one that came into my heart and mind at that point was, wait a minute, maybe we are the exquisite instruments into this field, human beings. Maybe the human being is the greatest instrument ever created by this very process to sense this field. So the people who are working on all the technical parts of the science are using their minds to go into the field, and they're using science. But the people who have deep and profound and intimate connections with the world or each other, or something we haven't even figured out, are also exquisite instruments of science or of understanding. We are the best instruments we'll ever make. Human beings, humans' bodies, minds, and hearts are the best instruments, potentially, to work on this thing. So with our cells and our tools, we can explore this field. So in a sense, the dream was inviting us to keep exploring the field. Don't name it so much. Be open to what it teaches you. Learn to speak a common language amongst ourselves so that we don't get all tripped up. And that one day we may come to a more full knowing of it. But this is a process. This is part of our evolution. But we're exquisite instruments to do this job. So I want to leave you with this. Everybody presents models that maybe can be possibly worked on, and maybe you guys can work on. They call this rotary roadmap of consciousness or to consciousness. So in this model that I presented to you, the universe is a cycling system. It has to cycle, it has to turn. Stars are born, they explode, they reform, they explode. But it tends to be two phases, like a two-phase motor, like a lawnmower. It can mow the lawn, but not like a three-phase motor that can do three-cycle motors. So it doesn't have memory, right? It doesn't have a memory. So in our work with our colleagues in physics, we're literally coming to the point where information in physics is more binary. It's not linear, it's not a memory. But we're now, through self-assembling polymers and testing them, we're finding memory in physics, but it steps into toward biology, found that boundary. So you're going from two phases to three. But that system works for billions of years such that the three-phase cycle can emerge, which has memory. It has bodies. It has an intermediate phase, the progenote, the little sludge. Intermediate phase, it can pick things up, it can learn, do all sorts of things, learn chemically or innovate. And that animates matter into living systems. Then you get to neural dynamics. And that's a big transition, perhaps. That's the fourth phase in this cycling system that's using all the previous phases. It's like stacked upon stacked upon stacked. And what that means is you can have learning in a system. You can have goal-directed behavior. That's a new technology. A paramecium can do it ostensibly with just chemical circuits. But when we get nervous systems, we can do it much better. But that's a new phase. How about consciousness? Is this a fifth phase that the whole thing goes through? Dick, 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 dick. Is there something there? Because we know those previous phases work. We can characterize them. We can study them. That fifth phase, what is it? What is it? What is it? That's your department, perhaps. So I want to thank you for your attention. And these are our collaborators on the Origin of Life work. And if you have Scientific American, you'll see our publication in July or August, which will introduce it to the general public. Thank you very much.
Fantastic. That was worth waiting for Saturday afternoon, for sure. Thank you, Dr. Damer. So are you enunciating an imperative for the human species to travel beyond our Earth?
Well, I think I dig it.
Yeah.
I dig it because I
Well,
think,
you work for NASA, right? Come on.
Yeah. I like it because it gives us immense perspective.
Yes.
So for instance, if we're able to, it takes the pressure off the biosphere too, because a living system is just trying to bust out, and we're potentially the reproductive organ. So Gaia is saying, yeah, take it all, as long as you do the act and the messy act of cell division, right? So cell division's messy. Birth, as all women will know here, it's a messy process, but it's good because you have another. And so if we are the mechanism for Gaia to go forward, because in 100 to 200 million years, the sun's on a heat curve, and the terminator's approaching, right? This is in Lovelock's last book. Venus was gobbled up by heat death. I mean, atmosphere left and went in the atmosphere, and horrible, it's 900 degrees on the surface of Venus. That's coming to us. And Lovelock's calculations are it's 100 million years out. So when that happens, we go to runaway greenhouse, no matter how much or how little CO2 we have in the atmosphere. So it's over for plants on land, it's over for animals. Eventually the oceans will be gone. Microbial life will continue all the way to the heat death of our planet. But we're at that 1%, 1 to 2% of complex life that, say, a planet like this can sustain. We're coming to the very end of it. So if Gaia exists as some kind of a force of nature, she's like, we got to do this thing. And what that means is getting life out of the gravity well and creating other biospheres. And so Shepard, the design of Shepard can do that because it can encapsulate a large asteroid, melt it down to a rock. We can introduce biology, and we can feed people in space and construct stuff in space. It's a sailing ship invention for space.
It's our imperative to survive. A question here. We're going to take this one first.
What can you say about the implications of your work for the Fermi paradox?
Now that's the thing that they should be out there, but we don't hear from them.
Yes, that's right. Yeah.
We're all little green men. I think I did a talk at the SETI Institute where I added 4 new terms to the Drake equation, which was the to boldly go terms for Star Trek, and Seth Shostak was there, so we had a good laugh. But it's so extraordinarily rare. Technological civilizations have done what we've done. It's just so rare. So I wouldn't be surprised if they're separated in time and they're separated in space, and just never contact them.
Next one.
The reason why this is relevant is because if we make life more likely to happen, to emerge complex life, then we have to account for why we don't see more complex civilizations in the universe. So how much of a warning is this for us to pay attention to that
I
issue?
think what this is, as we look back and with the best science we can do, and we find out we're so special, not in church ladies special, right? But we are incredibly special. We're incredibly precious. The earth is precious. We are precious. We really have to take care of this place and each other, and just like, wow, look at, we're here. And and we have a responsibility because of all the effort. I sometimes have conversations with these guys, and when I'm doing mental activity at the progenote level, I say to the smudgy progenotes that are booting up to life, I say, guys, it's going to work, we think. Mars, it didn't work out. We're working a little bit of overtime. I don't tell them about all the stupidity that humans do. But I say, work the overtime, clean our atmosphere, become life, clean our atmosphere, do all the things, because this one might pan out. And they go, yeah, right. But in a sense, this is your ancestor. This is your ancestor. And we owe it to this system, having persisted, whether through will or just through just doing it, just cycling, to be, in a sense, honor the incredible labors that this ancestor did, so we could be sitting in a room breathing oxygen and eating the ancestors for lunch, and making technology, and then asking, and then looking back at them, finding them. It's an amazing moment.
Next question.
This is a fascinating talk, but for any of this elementary algorithm or whatever you call it to work, you need two things. One, you need energy, which is okay, we have them. But what's really needed is a mechanism for the entropy to decrease consistently. A consistent way for the entropy to decrease. Where is that mechanism?
Through wet drying, wet-dry cycling. So
The wet, dry cycle.
wet-dry cycles.
How would the entropy be decreased?
So you're going, what happens? You have these films floating in the water, and you have building blocks, monomers. The system dries down, goes from a three-dimensional medium to two-dimensional, and then evaporation occurs, taking all that entropy out. And then between the films, the building blocks line up, and then water leaves, and they go click, 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 and form a structured polymer, which nature does with ATP. Nature kicks water out with ATP. And so that's where your system undergoes that transition.
Positive feedback?
Sorry?
Some positive feedback mechanism?
Yeah, and then it cycles again.
Okay.
It keeps going and going and going. And that's why we're elongating and growing more polymers away from equilibrium.
So I've read that part that you just explained. Peptide bonds don't form well in aqueous solutions. They get destroyed by hydrolysis. So is that how you're saying that works?
Yes.
Yeah, okay.
Exactly, yeah.
Oh, yeah, go ahead.
And a related question, Jerry Pollack's work with fourth phase of water and ordering, he talks about the exclusion zone being set up by optical pumping. Would that play a role also in the entropy reduction, perhaps, that Dr. Chua was just asking about?
It's possible. We don't need it at the moment. We're dealing, it's possible. And quantum effects could be possible in this epoch too, because ultraviolet light, we think that the first pigments were polycyclic compounds, and that would involve some kind of quantum effect to turn that into an energy source, because this early life can't stop just being dependent on the fast food outlet of the hydrothermal field. It eventually has to make its own food. And that's a huge transition for the progenote world to do photosynthesis. And it's not going to be with chloroplasts, it's going to be with something much simpler. And that's where some of the quantum effects might come in.
Quick question, two last quick questions.
Okay. First one's just a comment. If you're curious about the Fermi paradox, Google "They Are Made Out of Meat." It's a great little movie.
Okay.
Yeah. Because they're not here because we're made out of meat, and that's what intelligence is not made out of anymore. It's all made out of metal and silicon. Anyway, I've had this argument with several creationists who make the point that if you take the time at which the sun first started to fuse, and the time at which we have evidence of life in the rocks, it's a very short time. It's a surprisingly short time. And they make the argument that it's too short. But I've read a couple of papers that say that it's quite possible that protogenotes may in fact evolve in the interstices in rocks, essentially asteroids. Have you encountered that argument? And if you have, what do you think of it?
The chemistry only takes you so far, so the building blocks are going to be forming in the solar system accretion disk, especially as the sun heats it. And then they come in the atmosphere. Now here's a fun one for you guys. We can now do the numbers on this thing because of what the comment was back here about peptides breaking down in water. So we know the outer bounds of which we make a nice peptide, it gets encapsulated, it cycles back. It's got to do it in probably hours to days. There's no refrigerator for us to put our stuff in. So we have outside bounds. We have a box. Like progenotes have to cycle at X rate, and it's hours to days. Might be minutes, maybe 23 minutes like Old Faithful, or 73 minutes filling and et cetera, or through dew cycling, through day-night cycling. It's fast. So we can get to progenotes, potentially something that starts to evolve and adapt and then fails many times and in many places on a ancient Earth. And so there can be starts and failures, starts and failures, starts and failures, starts and failure. And how we get over that, the big gap, how we cross those huge evolutionary gaps is that something started inventing something cool, but it's dying because of all the waste product buildup. Wind comes and blows a piece of it to the next pool and shares that innovation with something else that has something else. We have horizontal gene transfer, the roots of the tree of life. So that system can stay ahead of its ricketiness. You know, and a huge asteroid hits the Earth, washes an entire island over with a 1,000-foot tsunami. That's fine. The cataclysm will wash the system out and allow the few remaining protocells to start again. So this is why I don't believe in panspermia, because, you know, life is so hand in glove with this planet. It grew up with it. It changed the planet. It created new minerals, for goodness sakes. We create minerals. You know, our cell phones are phonite. You know, our brain case is a mineral that contains a liquid crystal mineral called the neural structure. And one last point for you guys. So I'm standing around with geologists, and I said, okay, let's do a thought experiment. We'll build a time portal. You can walk through it and go to 4.3 billion years with your spacesuit and your environmental suit and everything. And we'll go to these pools. And we go to the pools, and we see the sludge in the bottom of the pools. The geologists would say, that's just sort of a mineral fusion sludge. The biologist said, that's kind of interesting because it's kind of growing. So we come back 100 years later, and the sludges are in all the pools, and some of them have turned black. And the geologist said, minerals don't turn black on their own. Well, and the biologist, let's pipette that thing, put it in our sequencer. Oh, there's a little short oligo of some RNA-like thing or DNA-like thing, and there's a lot of them in there, and they're making black pigment to shield the whole community collectively and introduce photosynthesis. So this is where geology and biology separate. Geology used a hard minerals to make their forms. Biology used soft minerals that could learn how to make their own solutes and keep growing and building. And here's the last piece for you guys. It's sort of an extension of the talk. Here's the beautiful thing for philosophy from this. We didn't start from competing protocells. We started from a collaborative community unit. It's a network like the internet, where every part is contributing to the whole. This could be proved by science in less than 20 years, because we could be growing these things in science, and we'll see them fill the dishes, and then if they crash down, like if you do time lapse, and they grow again, they've responded to stress, we pipette that, and we find that they've undergone molecular evolution. There could be competitions all over the world to do this protobiology, but it'll be like seeing the Earth from space from Apollo 8. You know, that beautiful image? Seeing this is probably how we came into being. And it's a struggle. It's like go team go, you know, because these things are fragile, they fail all the time, but they're collaborating and they're reaching up. So when we hit them with hard ultraviolet, they respond and they turn black. We go, wow, this is our ancestry. We'll rethink our civilization at that point. Certainly our political economy thing, we should. So anyway, that's where this rolls into philosophy.
Very last quick question.
Well, I was going to ask the question, what was the first truly self-replicating thing? But I think what you're saying is there was no such thing, that there wasn't an individuated self-replicating thing. There couldn't have been. That's a stupid question to ask.
And that's where it goes to creationism again, because they talk about this as being the irreducible thing. Well, you don't have to have it. So the little bundle of things, the bundle of molecules when I was 14 that winked, like, you work on it. When I saw it in December of 2013, I was doing yoga and breath work, and I went into a reverie, and I was the protocells. I was just 2.5 hours, I was running the thought experiment. I ran upstairs, wrote to Dave Deemer, my colleague at UCSC, did the drawing, and he wrote back, you found it. You found the kinetic trap. You found the mechanism. And the beautiful thing about that is it's solved. The wink was solved. So I could say back to the dancing molecules, it was little machines all working together that lift in an environment. And out of that environment, that niche, that could come more complex machines that one day could divide on their own. But they did it only within the context of the community to support them. the whole thing. That's the whole basis. You can't do things without a community support around you. And that could be shown at the protocellular level.
And you heard it here first. Thank you so much, Dr. Damer.
Slow ahead. I can go slow ahead. Come on down and chum some of this shit.
You're gonna need a bigger boat.